The 22% RNA failure rate. AlphaFold 3's latest April 2026 benchmark data shows a persistent failure rate of 22% when predicting RNA ligand interactions, despite its dominance in protein-only folding. While the marketing materials focus on the phrase "the prediction of life," the actual architecture is a specialized diffusion model that moves away from the sequence-heavy logic of its predecessor. The core challenge is no longer just folding a string of amino acids, but simulating how those folds physically crash into DNA, RNA, and small molecule drugs in three-dimensional space. We are seeing a transition from pattern matching to a form of statistical physics that prioritizes atomic distance over evolutionary history. The death of the Evoformer. In AlphaFold 2, the Evoformer block relied heavily on multiple sequence alignment, or MSA, essentially looking at how nature solved a folding puzzle in the past to guess the current structure. AlphaFold 3 strips much of this away, replacing complex MSA processing with a much simpler pair representation system and a heavy-duty diffusion module. This is a technical gamble. By reducing the reliance on evolutionary data, the model can finally attempt to predict synthetic molecules that have no evolutionary history, like new plastic-eating enzymes or experimental chemotherapy agents. However, this shift means the model lacks the evolutionary guardrails that kept previous versions from making basic geometric errors. It's more flexible, but it's also more likely to hallucinate a bond that is physically impossible. The small molecule bottleneck. Modeling a ligand is fundamentally different from modeling a protein because small molecules don't follow the modular rules of amino acids. Think of it like this. A protein is a chain of 20 standard Legos, but a ligand can be any combination of atoms in the periodic table. AlphaFold 3 handles this by treating every atom as a point in a diffusion cloud, ignoring the chemical bonds initially, and letting the model learn the physics of proximity. It's a brute force approach to stereochemistry. The problem is that the model often ignores chirality, the left-handed or right-handed orientation of a molecule, which is the difference between a life-saving drug and a toxic compound. In the May 2026 validation sets, nearly 1 in 5 ligand predictions still exhibited these handedness errors. The data scarcity problem. DeepMind's training set for proteins is massive, thanks to decades of global research, but the data for DNA-protein interfaces is several orders of magnitude smaller. This creates a massive imbalance in the weights. While the model can predict a protein's alpha helix with high confidence, it often stumbles on the flexible loops of RNA that are critical for gene expression. The model isn't actually learning the laws of physics from first principles, it is approximating them from a biased dataset. This is why we see high performance on rigid structures, but a sharp drop-off when the molecular interaction involves water molecules or metal ions, like zinc and magnesium, which the model often fails to place correctly within the binding site. The shift toward molecular dynamics. The move from AlphaFold 2's evolutionary lookup to AlphaFold 3's diffusion mechanism marks the end of simple structure prediction and the beginning of actual biochemical simulation. By focusing on the position of every atom across DNA, RNA, and ligands simultaneously, the architecture is effectively attempting to bypass the need for traditional molecular dynamics, which usually takes months of supercomputing time. This shift toward diffusion-based modeling means that the bottleneck for drug discovery has moved from finding the shape of a target to verifying the stability of the binding. For researchers, this means the model is no longer a final answer but a starting point for high-fidelity physics simulations. The real signal under the noise isn't that AI can fold a protein, but that it can now predict the docking energy of synthetic molecules that never existed in nature. Watch the upcoming CASP-17 results in late 2026 for the first independent verification of these multi-chain interactions in blind tests. Subscribe for more.